So here we move for digestion for part 7 for grade 8. So we talk about the activity number 4. Digestion is divided into mechanical and chemical. So mechanical is going to digest the food into smaller pieces and will keep the food moving through the long digestive tube. If you remember, digestive tube includes the mouth, esophagus, stomach, small intestine, large intestine, and finally the anus. So mechanical is the mechanical action. It's breaking down the food into smaller pieces. So this is the mechanical digestion. However, the mouth we have in the mechanical digestion is called chewing. In the stomach, we have churning. In the small intestine, we have peristaltic movement. So these are the three different types of mechanical action. In the mouth, we have chewing. In the stomach, we have churning. And in the small intestine, what do we have? Peristaltic movement. You have to know this. Chemical digestion. What is the chemical digestion? It digests the food by the help of digestive juices. As we said before, digestive juices, they contain what? They contain enzymes. So these enzymes, they are very important for the process of digestion. So they are found where? In the digestive juices. For example, it breaks down food into simpler nutrients by the help of these juices that contain the enzyme. For example, in the mouth, what do we have? We have the saliva. Saliva is the juice that contains which enzyme? Salivary amylase. So this enzyme, salivary amylase, it works on what? On starch. To give what? To give maltose. The medium is what? The medium is neutral and the pH is equal to 7. This is at the level of the mouth. At the level of the stomach, what do we have? We have the enzyme pepsin. Pepsin is found in which juice? The gastric juice. And it works on protein to give peptide. The medium is acidic, meaning that the pH here it's less than 7. Well, here the pH is greater than 7. No, it's equal to 7 or approximately. If you want, approximately it's equal to 7. And in the small intestine, we have the pancreas and we have the small intestine. On the pancreas, we have pancreatic juice and intestinal juice, and the medium is basic, meaning that the pH is greater than 7. It's greater than 7, yeah, means that's greater than 8, 9, 10, 11, and so on. Here, chemical digestion and mechanical, both of them, they are needed. How? We said that the mechanical digestion, it cuts the food into smaller pieces. So in order to eat the food, it should be cut into smaller pieces. So mechanical digestion, what does it do? It facilitates the chemical. What does this mean? This means that it will help the chemical digestion. So mechanical cutting the food into smaller pieces will make the digestion easier by the help of the enzyme, which is the chemical digestion. So mechanical digestion will facilitate. What the meaning of facilitate? Will make it easier and will make the digestion easier. So here, mechanical digestion, cutting the food into smaller pieces, will help the chemical and allows digestion to finish faster. This is what's known by the mechanical digestion and the chemical digestion as well. So this is the important deduction or conclusion. Mechanical digestion facilitates the chemical and not the vice versa. So cutting the food into smaller pieces, whenever you eat small piece of meat, it will be digested faster than eating large piece of meat. Why? Because small pieces that are digested faster, since mechanical digestion facilitate the chemical and allows digestion to finish faster. Moving forward, the root of the nutrients. Where are the nutrients? Where do they go? We have an important phenomenon that occurs at the level of the small intestine. This phenomenon is known by intestinal absorption. So intestinal absorption of the nutrients, it occurs at the level of the small intestine. Why does this process occur at the level of the small intestine? We have 4 important characteristics that makes this small intestine a good place for intestinal absorption. First important characteristic, number 1, thin wall of intestinal cells. What does this mean? We have very thin wall that allow the absorption of the nutrients. Number two, large surface for exchange. We have large small intestine. It's very long, and this allows for exchange. It make it easier. Third one, highly vascularized. What the meaning of highly vascularized? Meaning that it's rich in blood vessels and lymphatic vessels. So it's rich in blood vessels and lymphatic vessels. It's known as highly vascularized. Mister, can we study this without using this word highly vascularized? I will say, no. You have to know both of them. You have to know highly vascularized, which means that it's rich in the blood vessels and lymphatic vessels. Number four, the fourth property is the moisture. This will allow the intestine to be a good place and favors, meaning that facilitates intestinal absorption. So this is important characteristics. You have to study them that facilitate the process of the intestinal absorption at the level of the small intestine. Absorption of nutrients, it occurs in two ways, either at the level of the blood or at the level of the lymph. At the level of the blood, all the nutrients are absorbed, except what? Except fatty acids and the glycerol. What is the root for this? These are absorbed at the level of the lymph. So the lymph absorbs only fatty acid and glycerol. However, the blood, it absorbs all the nutrients unless fatty acids and glycerol. Moving now to the worksheet. So before I talk about the worksheet, we will finish everything related to digestion. The digestion chapter is almost done. So here I'm going to move to the first question. We're going to solve question number 1 in the worksheet. And the second question is very easy. So a student in grade 8 suggested that enzymes, they are biological scissors that break down the substrate under different pH media, meaning that under different pH, in order to test this hypothesis. Which hypothesis? This is the hypothesis. It's mentioned in the text. This is the hypothesis. The hypothesis is enzymes, they are biological scissors that break down food and substrate under different pH media. This is the hypothesis here. In order to verify this hypothesis, so here it's already mentioned in the text. Which hypothesis is the sentence before this? So in order to test this hypothesis, which hypothesis? Yeah, this one, enzymes till pH media. In order to test this hypothesis, trypsin enzyme is added into two different tubes under different pH media. Same conditions of temperature, convenient time duration, يعني a suitable time duration, and substrate, which is the albumin. Already we talked that albumin is a protein. So we have two tubes, C and D. Yes, Maya, your question is? Yes, Maya?
Yes, it says no. I just wanted to answer a question.
Okay, yeah, let's start step by step. Yeah. So first of all, we have, in order to test this hypothesis, in the first part, they ask you pick out from the text the tested hypothesis. What is the tested hypothesis? Enzymes, they are biological scissors that break substrates under different pH media. Okay, thank you, Maya. So this is the hypothesis. Pay attention to read the exercise very well and to know how to answer the questions. We have two tubes, C and D. Two tubes, they are placed where? In a water bath at temperature 37. Water bath, it's like a big beaker, contains a water, and the temperature is 37 degrees Celsius. Both tubes, they contain albumin. Both tubes contain trypsin. Both tubes, they contain water. So all these conditions, they are common. All of them, they contain albumin. All of them, they contain trypsin and water. All of them, they are placed in a water bath, temperature 37. But In tube C, the pH is equal to 2. However, in tube D, the pH is equal to 8. So what is the variable factor in this experiment? The variable factor is the pH. Since here the pH is 2, however, the pH here is 8. If I ask you, yeah, I want a new students here, new hands. If I ask you, Abbas, what is pH equal to? What does the pH equal to
Yes,
represent? Acidic, basic, or neutral?
it's acidic,
Acidic, pH equal 8.
basic.
Basic, thank you, Abbas. Moving now to, I need a new hands here. Ali Fadi Basma, good morning. Ali Basma, good morning. Ali Fadi Basma, can
Yes,
you hear me?
yes, Ustad.
Okay, Ali, trypsin enzyme, it works in acidic medium or in basic medium or in a neutral medium. What do you think? Trypsin enzyme, it works in acidic, basic, or
بس
in
بدك
neutral.
ترجع تعيد.
Trypsin enzyme, it works in acidic, basic, or neutral. Okay, what do you think, Kareem? Thank you, Ali. What do you think, Kareem?
Acid.
Mr. Muhammad, trypsin is basic because it works on the level of the small intestine.
Okay, the level of the work is more extensive, so it's a basic one. So it works in a basic pH. Thank you, Karim. So trypsin, it works in a basic pH. This one, these notes, I have to know them before I start. That's why I ask you to study the tables and to review them before this session. Because if you review the notes about the enzymes and about the food test, this will help you a lot. After 60 minutes, what happened? The albumin, it persists in tube C. So there is no digestion in tube C. However, we have this appearance of the albumin in tube D. So where did the digestion occur? The digestion occurred in tube D and not in tube C. Here I analyze and I explained all the figure and all the experiment before I start. Let's move now to the first part. First part, pick out from the text the tested hypothesis. Whenever they said pick out, meaning that I have to take it from where? From the text. So pick out from the text the tested hypothesis. So I have to go back to the test. Tested hypothesis. What is the tested hypothesis? Pick out. I put it between quotations. Enzymes are biological scissors that break down food or break down substrate at a different pH media. Only this sentence. اللي بيكتب أكتر من هيك, this means that you don't know what is the hypothesis. So بيقولوا الزايد خلي ناقص. In biology, if you add any additional information, this means that I'm going to remove the mark. So here only you have to write this. Enzymes till pH media. This is the tested hypothesis from the text. Referring to the figure, pick out the variable factor. So as I said before, all the conditions, they are similar unless the pH. pH equal 8, your pH equal 2. So the variable factor is only the pH. Moving now to part C. Justify the uses of water bath 37 degrees Celsius. Before, can all we use water bath, we use temperature 37 because it's the normal body temperature. Yes, but you have to add for this, it's the normal body temperature where the enzyme, they can work at the normal condition. So this is the best temperature for the working of the enzymes in our body. ما بس بنقول it's the body temperature. It's the best temperature in the body. It's the body temperature where the enzyme, they can work at their best. Represent in the same table the conditions and the results of the experiment, C and D. Some of you, they asked me, Mister, you asked us about Table of conditions. I don't know what is the table of condition. Yes, this year we didn't draw table of condition, but this was in the last year we drew a lot of table of conditions, if you remember, in grade 7 when we studied about digestion. So here table of conditions should be clear for all of you, and you have to know how to draw the table of condition. This means if you studied it in grade 7, this doesn't mean that we have also to explain in grade 8. Already is done. Here the table of conditions and also the graphs, they are like interpret and analyze. We have covered this before in grade 7. So this should be with you. So table of condition, this is the table of condition. First of all, table of condition, let's move for example parts A, B, C, and D together. So A, I pick it out from the text, pick out. Part B, sorry, what is the variable factor is the pH. Why do we use temperature 37? It is the enzyme, they have optimum function. What the meaning of optimum? We said the best function at 37, which is the normal body temperature. اللي بيقول لي بس it's the normal body temperature, this is not correct. بياخد العلامة. You have to say the enzyme, they work at their best at 37 since it's the body temperature. Moving now to the table of condition. Table of condition, first, you have to divide it into conditions. These are the conditions. And here the result. And here we have the tubes, C and D. What do we have? We have substrate. We have the enzyme trypsin. We have water. We have pH. You can say time or you can say duration if you want. The same. And you can add temperature and you can add the results. So albumin is present in both of them. Albumin, and you can say plus. What does plus means? You have to use the legend. If you didn't put the legend, this means that you won't get the mark of the table. So إذا حطينا plus, شو يعني plus? Plus means present. Okay, so you have to say it. This is called the legend. Remember this from the last year. Legend plus, it means presence. Plus, water is present in both of them. So plus and the plus. pH, it's 2 in tube C and it's 8 in tube D. Duration, it's 60 minutes in both of them. Temperature, it's 37. Results, albumin exists or remains or stays the same. Disappearance of albumin in tube D. What I have for the table, I have also the title. Title, table showing the conditions of the results of the experiment in tubes C and D. So here, this is table, should be drawn using a pencil. Second, should be given a title. Third, as a handy legend, we have to use legend. Sometimes we use not only plus, we use minus. So if we use minus, it means the absence. So if you use the minus, this means the absence, you have to put it. Here, I didn't mention this one because I didn't use it in the table. So this is the table of conditions and results. It's from the last year. It should be done using a pencil. Yes, Kareem, your question is?
Ustaz Muhammad, can we put in the result minus instead of the disappearance of albumin?
Can you repeat? I didn't hear the part. Can we put in the results plus and minus? Did you say this?
Yes, instead
No.
of disappearance and
No.
exists.
No. You can't put plus and minus here. No. And the results, no plus and minus. This is not accepted. No. Okay? You have to say in the results, you have to write them as a text. Persist or exist or remains the same or disappear. Plus and minus is not accepted. Even though, if you put this means that you are talking about conditions. The result should be written in the form of a text. Okay, Kareem?
Yes, Ustaz.
Okay, great. So let's move now to part E. They said interpret. Interpret, this is the big problem here. What is the meaning of interpret? It's analyze plus indication. So analyze plus indication. Interpret, it's analyze. I repeat for this for the 100th time. Analyze plus indication. So we have analyze plus indication. In the quiz last time, what did you do? Some of you, they did only analyze. I gave you, for example, if this interpret, it has 2 marks. I gave you 1 over the 2 since you did only the analyze. This time, if you did the analyze, you won't get any mark. Analyze is not accepted. If it's interpret, you have to write analyze plus indication. Other than this, it will not be accepted. Is the proposed hypothesis validated? It's verified. It's correct. If yes, you have to justify. So let's move to the answers of this. After adding trypsin and water to albumin in tube C in a pH 2 at temperature 37, the albumin remains in the tube after 60 minutes. What does this indicate here? I did analysis, but I finished, I put the full step, and I did the indication. This indicates there is no digestion of starch or digestion of albumin, sorry, in tube C. However, after adding trypsin plus water to albumin in tube D in pH 8 at temperature 37, so the albumin disappears. This is the analysis from here to here. This is the analysis, but this is the indication. This indicates that we have digestion of the albumin. So, proposed hypothesis that the enzyme did work under different pH? Yes. Why? Because when the pH was 2, we have no digestion. However, when the pH was 8, we have digestion. So the pH is the variable factor. So trypsin, it works on a protein to give what? To give amino acid. So the final product will be amino acid. So how can we verify this? We can do violet test. If digestion, it doesn't give a violet, so we have digestion of proteins and or polypeptides, it occurs. If we have violet color, meaning that the proteins and or polypeptides, they still present in the tube. So here, pay attention to put the variable factor in the analysis. If you didn't put the variable factor, you won't get the full mark. Interpret, analyze, this is the analyze, and this is the indication. So pay attention to this. Review also the table of enzymes, the table of results as well, and you have to review the notes related to the food test and their positive and negative indications.